All right, good morning, everyone. Look at what we're doing. I am doing the chapters that I said you would have to read on your own over vacation. Look at me stepping up and being morally supportive while we're all bored out of our minds. All right, so we're still going on with the unit on evolution. And now we're talking about populations. Because one of the things we left off with last time is that, remember, individuals do not evolve, populations evolve. I know, it was like a lead-in to the next chapter on population evolution. It's good like that. Oh, see? It's twice. I said it, they said it last chapter, it's right there, okay? Natural selection acts on individuals because those individuals best suited for the environment are able to reproduce more. Therefore, what? Their genes get passed on. So, talk about Darwin's fishes. Oh my goodness, we're done with finch. Oh, there's a finch. Look at how cute. Oh, there's a graph. Beak depth. We'll keep moving on that. So, it's talking about no one is gonna ask you a specific question on how the beaks evolved. Know that the beaks evolved in order to fit in the correct situation that they're in. So in other words, if they were seed eaters, it developed to better feed off seeds. If they were going into nectar, if they were going into a particular flower, if they were insect eaters, you could see how that would work. So, microevolution. This is where we get down to it. It's the change in the allele frequencies of a population over generations. Okay? The change in allele frequency. That's what's important for you to know. So, 3 mechanisms are causing this, and we need to know those. You need to know the difference between natural selection, genetic drift, and gene flow. And only natural selection consistently causes adaptive evolution. So, obviously, we'll go through those 3. Variations in heritable traits is a prerequisite for evolution by natural selection. Unless, we go back and we start talking about bacteria again, but we're not. Let's kind of stay with the theme of the chapter. So Mendel's work on pea plants provided evidence of discrete heritable units. Remember, Mendel did all this stuff without even knowing the concept of genes. That's why you'll sometimes don't get confused between the term allele, gene, allele, gene. They're basically the same thing because Mendel didn't know anything about genes. So phenotypic variation often reflects genetic variation. We're aware of that. This is nothing new. Thank goodness. Look, we've made it like 3 slides and haven't had to learn anything new. So, generic variation among individuals is caused by differences in genes or any other DNA sequence. We went into, and you guys can remember, different DNA sequences that cause different traits, whether we're talking about genetic disorders that you're going to have to pull out. Always what? Sickle cell, keep that in mind. Okay? Some phenotypic differences can be classified as either or. They're usually determined by a single gene. Other phenotype differences are due to the influence of many genes and varying graduations along the continuum. We talked about that with epigenetics. We talked about that with skin coloration. Okay? And there are a bunch of different horses, in case you didn't know what a horse looked like. Okay? It can be quantified as the average % of loci that are heterozygous. Yeah, no one's ever going to ask you that. Let's just keep moving through that one. It can be measured at the molecular level of DNA. So this is where we're doing it now, okay? So this is where scientists are going and looking at the amino acid sequence to see how different organisms vary. Nucleotide variation rarely results in phenotypic variation. We know this because we learned about our codon sequencing, okay? We learned about the wobble, whereas the 3rd one in the codon usually has nothing to do with anything. So that's what they're talking about there with nucleotide variation. Most differences occur in non-coding regions. That seems weird on introns, things that stay in that don't get expressed. Variations that occurred in coding regions, exons, rarely change the amino acid sequence of the encoded protein. Because if you're a human, pretty much all humans name the same stuff. Here are some examples of some substitutions, base pair substitutions. We remember about transposomes. We remember about insertions, deletions, whether we're talking about on the nucleotide level, whether we're talking about on the amino acid level. We can talk about frame shifts. You may have to. Cat helping out, just saying. Okay? A phenotype is the product of both inherited genotype and environmental influences. So if you're, say for example, if we're talking about people and you had two twins and one grew up in a really nice neighborhood where they had all the food, all the medicine, everything they had there. Another one grew up in a poor environment, say on the other side of the planet, I don't know, in a country where they don't have a supply of food, they don't have a supply of water, they don't have access to medicine. Those people are going to grow up into vastly different in the phenotypic direction adults. Oh, look, two different caterpillars. Same caterpillar, same type. No one's gonna ask you to, you're never gonna see that picture again. Oh, here it is closer. You know it is closer. Okay. So, sources can result from the production of new gene and alleles through mutation, the original source, okay, of genetic variation, or gene duplication, sexual reproduction, meiosis. Go back, learn meiosis. If you didn't get it the first time, please make sure you go back and check that. Also results in genetic variation through the recombination of existing alleles. New alleles arise. I feel like I just said that. It's morning. I didn't even have a cup of coffee to start this omelet. So, new alleles arise by mutation, a change in the nucleotide sequence of DNA. Okay, so now they're telling us what a mutation is. Thank you. Only mutations in cells that produce gametes can be passed on. Well, yeah, if you get a mutation in your skin cell, you're not giving that off. It has to make sure that it's going to show up in, look, I feel like I'm at school, like random noises are distracting me. Mr. Jones coming in to, announce that we have a code red or something. Should we practice that at home? No, we're good. Okay. A point mutation is a change in a single nucleotide. Look, this is a bunch of redundancy, so I'm going quickly. The effects of point mutation can vary. Yes, it depends on where it is. So, mutations in non-coding regions often result in neutral variation. It doesn't matter because guess what? It's not coding. Okay? Mutations in genes can also be neutral because of redundancy in the genetic code. There we have our wobble. Remember we had, when we were looking at our amino acid sequence, what the first one was, what the second one was, what the third one was, then you had to go to the box and look at the line. Sometimes the boxes were bigger than others that coded for specific amino acids. So there's a lot of redundancy in the coding sequence. The effects of point mutation can vary. They alter the phenotype, are often harmful. Mutations that result in a change in protein production are sometimes, though rarely, beneficial. I'm guessing if they're beneficial, we could probably go back and look at it. Some of you people who are going to go study genetics can look at it backwards and see how that became advantageous. Chromosomal mutations that delete, disrupt, or rearrange many loci are typically harmful. Because from there on, you're changing, if you change your amino acid sequence, we know what happens then when you go to fold your proteins. It does not fold the same. So therefore, you have a different structure, which leads to what? A different function. Duplications of large chromosome segments is generally harmful, but small pieces might not be. Depends, once again, where this happens. So gene duplication likely played a major role in evolution. For example, an ancestral odor-detecting genes in mammals has been duplicated many times. Humans have 380 functional copies. Mice have 1,200. Wow, no one's going to ask you that. No one. Jeopardy? No, I don't even think so. Okay. The increase in the number of copies of these genes enabled mammals to detect faint odors and distinguish among many smells. They are smellers. Think, for example, taking your dog for a walk, which is probably the only way you're getting outside. Okay, taking your dog for a walk, how many things he sniffs, the things he smells that you do not smell. Oh, sorry, that is a cat because there's a bunny trying to dig his way into my house. Okay, so mutation rates are generally low in animals and plants, the average, and they can actually trace this. There are people whose job is to trace it back and look at how it affected evolution. I'm not kidding you, there are many jobs out there. Mutation rates are often lower in prokaryotes, but shorter generation times allows mutations to accumulate rapidly. Okay, this is probably how the first plasmid for antibiotic resistance came around. It was a mutation, but because they divide so rapidly, there we go. Now we have a whole new generation. They're faster than viruses. Oh, if this is not related to today, I don't know what to tell you. Because their RNA genome cannot be repaired by host cell, viruses are also able to accumulate mutations rapidly due to their short generation time. Well, yeah, there's not a lot to them. In organisms that reproduce sexually, most genetic variations result from the recombination of alleles. Most of it from the recombination of alleles. We also know that we have that crossing over in prophase 1. Please don't forget that. When we're going and making our gametes, okay, we don't know how our gametes are going to shuffle together. You know, is it this part of chromosome 1 and this part of chromosome 2 are going to make it into the egg or the sperm? We don't know. All that stuff gives you genetic variation. This is also telling you crossing over, independent assortment, and fertilization. All of those. Remember, we had the 2N, the number of chromosomes. So genetic variation is required for a population to evolve, but doesn't guarantee that it will, because once again, evolution takes time. Your genes are not like looking out going, hey, climate change is happening. I'm a polar bear. I better change, and when I have my next set of pups or baby bears, I better make sure that they get this tweaking of the genome. It doesn't work that way. Okay? One factor that causes evolution must be at work for a population to evolve. What is a population? We need to know what a population is, because if we're talking about that populations evolve, it's kind of good to know what a population is. A localized group of individuals capable of interbreeding and producing fertile offspring. Fertile offspring is hugely important. Hugely important. And it usually says in nature, okay? Because we've heard of like the liger and things like that, where you take tiger and lion DNA and you put them together. Number one, they can't themselves go back and either reproduce with a tiger or with a lion. So it's totally different, and they're sterile. So that makes them not, okay, a new species. Populations are not always isolated from one another geographically, but individuals typically breed with other members of their own population. Does that help you? Porcupine herd. That must be the name of those types of Artiodactylus, because those are not porcupines. I'm fairly certain. Oh, that's what it says, porcupine. We're thinking of two totally different things. So a gene pool is all the alleles for all loci in a population. An allele for a particular loci is fixed if all individuals in a population are homozygous for the same allele. So the frequency of an allele, and we kind of did that in a population, can be calculated. For diploid organisms, the total number of alleles at a loci is the total number of individuals times 2. The total number of dominant alleles at a locis is 2 alleles for each homozygous dominant individual plus 1 allele for each heterozygous. At the same loci applies for recessive alleles. Have we done this? I'm getting confused where we are. Oh, we might not have done. Okay. Hardy-Weinberg. I'm like, have we done Hardy-Weinberg? Why does it seem like it's familiar? Because I did it last year. Okay. Hardy-Weinberg, this is what we're going over with this one. By convention, if there are 2 alleles at a locus, P and Q are used to represent their frequencies. The frequencies of all alleles will add up to 1. So, P plus Q equals 1. If you're taking notes, write that down, because this is going to be part of an equation that you're going to need to know how to do the math. Okay, they're going to give you these equations. You're like, I can remember that one, but they're going to give them to you, and they even define these for you. Okay. We're going to go over there. We're going to go through this really quickly, because we're not looking at it like this. It's like, ah. So here it's talking about flowers and their genetic makeup. Remember, genotype, phenotype. Genotype, phenotype. We did Punnett squares, okay? But this is just going over and doing calculations at a big level. So P is going to stand for the dominant allele. Q is going to stand for the recessive allele. P is going to stand for the dominant. Q is going to stand for the recessive. okay? And the number of alleles in a population is always 1. It's like 100%. This is where I could use a chalkboard, okay? So, Hardy-Weinberg equation describes the genetic makeup expected for a population that's not evolving. This is huge, guys. When we do Hardy-Weinberg, okay, if we ever get together again, it's the best lab ever. If not, sorry, I don't know what to tell you. The best lab ever will not be done. So, Hardy-Weinberg describes populations not evolving. So you're going to have to know the rules that are involved for Hardy-Weinberg situation. And when you look at them, you're going to be like, that never happens. Yes! Okay, it's like a theoretical calculation. So the data observed for the populations differ from the expected values, then the population may be evolving. So if it doesn't come out to what Hardy-Weinberg says, because Hardy-Weinberg says in a non-evolving population, then you go with the assumption that your population is evolving. Okay? A population with mating is random, and none of the mechanisms of evolution are acting is Hardy-Weinberg equilibrium. These are the ones we're going to have to know, and I think there's 7, if I remember off the top of my head. So, allele and genotypic frequencies remain constant from generation to generation for a population in Hardy-Weinberg equilibrium. Mendelian inheritance preserves genetic variation in a population. Just throwing that in there. Okay, so here they're going through, and please don't get caught up on these numbers because they're awful. There is, go to Mr. Anderson Bozeman's Hardy-Weinberg. He does fantastic, and he has a chalkboard. I'm just using my TV, so you got to work with me. He's got a chalkboard or whiteboard or whatever, you know, where he's going to show you how to do the math. All right, maybe I'll put the direct link right underneath this lecture so you can go and see Mr. Anderson's Hardy-Weinberg and how to do the equations, because you will have to do them. They will give you the equations, but you have to be able to plug and play. Okay, you have to be able to plug and play. So Hardy-Weinberg equilibrium describes the constant frequency of alleles in a gene pool. And this goes through, and it's not necessarily clear. You know I'm not really good with graphics. So, okay, that we can kind of get. Here's something I'm going to point out to you. They're going to point it out to you on a slide better. P squared, okay? P squared, because we already have the equation. You wrote it down because you're paying attention and you're writing stuff down. P plus Q equals 1, okay? Ready? Let's do some math, or let's pretend. Say you square that whole thing, okay? So P squared, that is going to represent homozygous dominant. Once again, P squared is going to represent the homozygous dominant. Look at all the capital letters in there, okay? 2PQ is going to represent heterozygous. Q squared is going to represent homozygous recessive. Once again, if you take P plus Q equals 1 and you square it, you get P squared plus 2PQ plus Q squared equals 1. You're going to see that coming. But just so you know, P squared is your homozygous dominant. 2PQ is your heterozygous. Q squared, your homozygous recessive. And yes, you need to know that. Even if we don't do the lab, I'll figure out a way to get the lab to you for you to at least look at. Okay? Holy cow. You look at that all you want. I'm going to move past it because you know things like that give me little eye twitches. And more eye twitches. I can't even. So here we go. If P and Q represent relative frequencies of the only two possible alleles, remember, P plus Q equals 1. That represents the alleles. P squared represents homozygous dominant. 2PQ, heterozygous. Q squared, the homozygous recessive. Alleles, what they're going to show up in the population, okay? Please keep that in mind. Where P squared and 2p represent the frequencies of the homozygous genotypes, and 2pq represents the frequency of the heterozygous. So here it is, they're saying it out with that example that they're using with the confusing flowers. Why they couldn't have done it with something simple like Mendel's peas, I don't know. Okay, so Hardy-Weinberg equation. So now you have Hardy-Weinberg equilibrium, okay? You have Hardy-Weinberg theory, you have Hardy-Weinberg equation. I use the terms, I mess them up all the time, but the equation is the actual one with, you know, p squared plus 2pq plus q squared equals 1. So in real populations, alleles and genotypic frequencies do change over time. Yeah, we haven't reached equilibrium in any population. There's always something happening. Okay, oh, 5. So here are the things that you must know. You must be able to vomit them back on a test, pick them out of multiple choice, explain them on an FRQ. Okay, look at a population and they give you a population and you explain which one of the things are not being met as to why it cannot be in Hardy-Weinberg equilibrium. Okay, no mutations. Really? None? No, none. Otherwise, not this. Random mating. Random mating would be like pollen and a tree. Okay, it doesn't happen in mammalian populations. Okay, birds do good dances. If a bird sucks at his dance, he's not getting any birdie sex. We already know these things. Think about with people. Gosh, you can't think about people because number 1, ick, and number 2, we have no evolutionary sense anymore as to how we mate. Okay, no natural selection, extremely large population size. That's very important. You must have a large population size, otherwise you get skewed results. No gene flow. What does gene flow mean? That means no migration, no emigration or no immigration. Immigration movement into a population, emigration moving out of a population. Okay, we don't have any of that. So all of those conditions must be met in order for Hardy-Weinberg to be in equilibrium. Ain't gonna happen. Natural selection can evolve at some loci while being in Hardy-Weinberg equilibrium at the other. Some populations evolve slowly enough that evolution cannot be detected. So, and this gives us an example of PKU. The gene mutation rate is really slow. They can actually calculate it. Mate selection is random with respect to whether or not an individual is a carrier for it. Why? Because you don't know. Okay, natural selection can only act on rare homozygous individuals who do not follow dietary restrictions. Okay, you can figure it out. Little babies, you know, when they figure out they have it, all they do is change their diet. Okay, not like everyone's not changing their diet for something nowadays. The population is long. Migration has no effect, as many other populations have similar allele frequencies. So they were just giving you an example. Here they're doing some math for you. Okay, 1 per 10,000 births, which is this many. So that means you take the square root of it, you get this. And we know P plus Q has to equal 1. So that means Q, which is P, which is the rest of the population, has to be that. So the frequency of carriers is this. 2pq, because you have to include the heterozygotes in that, let alone the q squares, which are the expressors of it. Right? I know, we have to remember other chapters as we go on. Okay? 3 major factors alter allele frequencies and bring about most evolutionary change. We have our natural selection, genetic drift, gene flow. And obviously, these will have their own particular slides. Okay? Natural selection. Differential success in reproduction. Certain traits being passed on. We already talked about the long neck giraffe, okay? You can throw it around for anything. For example, an allele that confers resistance to DDT increases in frequency after DDT was used widely in agriculture. Okay, we can talk about the insecticide treadmill. Same like antibiotic-resistant bacteria. Okay. Then you have genetic drift. The smaller a sample, the more likely it is that chance alone will cause deviation from a predicted results. Oh, we're starting talking about chance. Oh no, do we have to do stuff? Could they do stuff with this and then go in and start asking you questions about chi-square? Yes, they could. So genetic drift describes how allele frequencies fluctuate unpredictably from one generation to the next. Genetic drift tends to reduce genetic variation. Genetic drift tends to reduce genetic variation, okay, especially in small populations. So we'll go through that kind of descriptor. Okay, you know, we're not doing that. Oh, look, more pictures. Okay. Showing you something. You look at plant pictures, you know I'm not good with plants. Here they are bigger because you know you like them. So, founder's effect occurs when a few individuals become isolated from a larger population, move to a new island. When people were colonizing different areas, they moved as a group, and they had certain traits that moved with them, because usually when groups move, similar groups move together. Allele frequencies in a small founder's population can be different from those in the larger parent population due to chance. Okay, here, let me go back. A good example of founder's effect, okay, I know it's going to sound a little weird, but up in Pennsylvania and Ohio, we have, in our population, we have a large Mennonite, which are offshoots of, I'm sure I'm saying that politically incorrect, of the Amish. And they interbreed, okay, that's also a wrong term with people, but you get me, right? They're all within their small population, and they stay amongst themselves, and they marry amongst themselves, and they have children amongst themselves. They have a huge proportion of children with polydacty. That means more than 5 fingers or 5 toes on each hand or foot. So that would be an example of something with a founder's effect, because they moved in there, so their population, that now became the norm compared to the parent population, which is generalized peoples. Then we get the bottleneck. It results from a drastic reduction in population size due to a sudden environmental change. An example of the bottleneck effect is what happened to cheetahs. They went down to so few that you had so much interbreeding just to bring the population back that one cheetah is almost a clone of another cheetah, is almost a clone of another cheetah. Okay? Or the Florida panther. Okay, that's closer. So here we have the original population. Here's coming out. Look, they're coming out. You only had that many. You should have had some yellow ones in there, right? Okay, no, I don't know if that's a good example for you. Okay. Florida panther, we know they're doing genetic inbreeding. How are they different from like the Carolina panthers? We know they have like a calic, you know, where your hair sticks up going in the wrong direction. That's how you can see physically. I'm sure they have other physiological. Oh, good. Bigger, 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 bigger. Okay. Understanding the bottleneck can increase understanding of how human activity affects other species. Sure, if we start isolating them, we start cutting off their habitat corridors, they can't change genetic, wow, that's a term I'm having. So in other words, they can't breed with anyone outside of their small area that they're stuck in because of things we've done, like, you know, 595. Okay, so it gives you an example, the greater prairie chicken. Okay, yeah, no one's going to ask you about the greater prairie chicken. There it is. There it is. Okay, there's some numbers. I mean, now you know. Okay, so they compared, we're still talking about the chicken. No one's asking you about the greater prairie chicken. Watch, that'll be like a big, huge question. Oops, okay. So, effects of genetic drift, the summary. Genetic drift is significant in small populations. Genetic drift can cause allele frequencies to change, can lead to a loss of genetic variation, and drift can cause harmful alleles to become fixed. So something that's detrimental now becomes the norm. So then we have gene flow. It consists of the movements of alleles among populations. They can be transferred through the movement of fertile individuals or gametes, for example, pollen drifting on the air. I mean, if it gets high enough up in the atmosphere, it can pretty much circle the globe, right? Gene flow tends to reduce genetic variation among populations over time. It can decrease the fitness of a population. It gives you an example. That, by the way, is a bird. Immigration of birds from the mainland introduced alleles that decreased fitness of the island population. Immigration from the mainland overwhelms the effect of selection. All right, we shall stop at that picture. We get to see it closer? We do. Okay, we'll stop here. We'll upload from there.